Okay. So just a quick overview of what we'll be talking about today. So an introduction to our VCEP, some background on Pompe disease and the GAA gene, which is what I'll be focusing on this time. And then going through some of our specifications. I'm not going to talk about every single one, but more of the codes that we've had some issues with, or just some more interesting discussion around. And then briefly just reviewing our ClinVar submissions thus far and our curation and review process. So just by means of an introduction to our lysosomal storage disorders VCEP, this is a subgroup of the inborn errors of metabolism working group. So that's our overarching clinical domain working group. We currently have about 15 active members, and I say about because our group has been around now for a few years, and people tend to come and go depending on the demands of their day job and so on. So I would say about 15 active members, including experts and biocurators, of course. And all our members at the current time are from the US and Canada. I will show the whole membership at the end of the talk, but just wanted to mention our fantastic co-chairs, who are Catherine Rader, who is director of the Diagnostic Molecular Lab at Duke, and then also Lauren Clark, who is a real expert in lysosomal storage disorders, has done a lot of research and followed patients over the decades, and he's at the University of British Columbia. So initially, when we submitted our application, we wanted to focus on genes for lysosomal storage disorders on the recommended universal screening panel for newborns in the US. Of course, as you're aware, the RUSP, as it's called, basically is this list of disorders that's approved within the United States for screening for newborns at birth. So we began by looking at the GAA gene for Pompe disease, and we're now just moving on to IDWay for MPS type 1. So the presentation today will be focusing on GAA because that's where the vast majority of our work has been done so far. So for some background on Pompe disease, this disorder was first described in 1932 by Dr. Johan Pompe, who was a Dutch pathologist. And he described a 7-month-old baby girl who had died suddenly with idiopathic hypertrophy of the heart. So she had this enlarged heart of unknown cause. And what Dr. Pompe noted was there was glycogen accumulation in the heart and other tissues. And I'm showing here an article that was written about Dr. Pompe, published in Muscle and Nerve in 2012, and I highly recommend this article. He just had an incredibly interesting life aside from his discovery of this disorder. He was actually responsible for sheltering Jewish people during World War II, and unfortunately was killed by the Nazis at the end of the Second World War. So really was a remarkable person in addition to his contribution to medical genetics. So Pompe disease is one of a number of lysosomal storage disorders. So at a very basic level, as of course you may know, the lysosomes are often known as the garbage disposals of the cell. So basically, lysosomes accumulate waste products, often macromolecules, and lysosomes contain many different enzymes that are responsible for breaking down these macromolecules in order to just maintain things are working well within the cell. Also to note, lysosomes have an acidic pH, so any of the enzymes working within the lysosomes will work at that acidic pH. So for lysosomal storage disorders, typically there is a deficiency of a particular enzyme activity that's required to break down one of those macromolecules. And then the molecules, of course, will build up in the lysosomes, causing this storage within the lysosome that can cause damage or dysfunction of particular cells and tissues. And for Pompe disease, the macromolecule that builds up is glycogen. So for that reason, Pompe disease is actually both a lysosomal storage disorder and also a glycogen storage disorder. So it's actually called glycogen storage disease type 2, as well as Pompe disease. And it has a frequency of approximately 1 in 40,000. I'll actually talk a little bit about the prevalence estimates a little later in the talk, but most of the literature will quote 1 in 40,000. Like many metabolic disorders, it's an autosomal recessive disease, and it is caused by deficiency of acid alpha glucosidase, historically known as acid maltase, although that term is really no longer used, but you will see it in the historical literature. So this enzyme is encoded by GAA. As I mentioned, it is a lysosomal enzyme, and it breaks down glycogen to glucose. And without this enzyme activity, of course, you get glycogen accumulation in the lysosomes of the heart and skeletal muscle and other tissues as well. So if you look in this figure here, this is actually showing progression of Pompe disease over time in muscle tissue. In this top figure, you'll see just these tiny little specks, which are showing accumulation of glycogen. And then over time, the purple is showing the glycogen. So this is PAS stain. So that's the accumulation of glycogen. And then you'll also see these vacuoles that appear as well. So just causing disruption to the muscle architecture over time. So in terms of what this does clinically, Pompe disease really is a vast clinical spectrum. So in the most severe form of the disease, babies are even born with already glycogen storage in their tissues. So the main clinical features of the condition are really profound hypotonia. So often known as a floppy baby, you can see here that this baby just really is extremely hypotonic. Also, a major component is this marked cardiomegaly, so massively enlarged heart, as you can see here on this chest X-ray, due to the accumulation of glycogen. And then over time, these babies typically die before their first birthday from cardiac and respiratory complications, and they can also have other issues as well. And then a little bit less severe, but still often with onset in infancy, we have this non-classic form where they typically don't have the cardiac involvement. And then there's later onset as well, which can have onset anywhere from childhood into the third or fourth decade of life, when you have progressive proximal muscle weakness, diaphragmatic weakness, respiratory failure, and so on. And these patients will also pass away after a number of years. But the late onset form tends to be much less specific features than the infantile onset form, and often presents like a lot of other neuromuscular conditions. So often what we see with late onset patients is that they have a huge lag in diagnosis from their onset of symptoms to actually being diagnosed. And often these patients, although they may not really describe onset of symptoms until later in life, when you talk with them, they may say, oh yeah, I was never good at sports at school, or couldn't really keep up with my friends, and that type of thing. But it really is just a continuous spectrum of severity. And there is a correlation between the residual enzyme activity in fibroblasts and the age of onset and severity of the condition, although there are some exceptions as well. So newborn screening for Pompe disease has been going on for, I guess, a couple of decades now. Enzyme replacement therapy for the disorder was FDA approved in April 2006 and has been approved in other countries as well. And the treatment is most effective if started early, before that damage is caused to the muscle tissues. So this was one of the, of course, reasons for recommending this disorder for newborn screening. And also it is possible to test newborn blood spots for the enzyme deficiency. So newborn screening occurs in various countries, including Taiwan and Japan, and also in over 20 US states. It was added to the RUSP in the US in 2015. So I'm showing here on the right an algorithm for newborn screening. So basically starting with looking at the enzyme activity in blood spots, sequencing the gene. If there are known pathogenic variants, then reporting this back to the family and seeing whether that baby has cardiac involvement or not. And then that will allow you to determine whether the baby has infantile onset Pompe disease, or may at this point have no symptoms whatsoever, but be predicted to develop symptoms later in life. And then we would also look at whether the patient is actually making any GAA protein, which would be CRIM positive, cross-reactive immunological material positive, or no protein, because that will help to predict whether that baby may have an immune response to the enzyme replacement therapy. So this was actually one of our sort of motivating factors for beginning this variant curation expert panel, was really to sort of help with this variant interpretation as part of newborn screening. So why is this important then? Like many disorders that I'm sure you all study and work on, Pompe disease has pretty extreme allelic heterogeneity. So while there are some common variants that I'll show on the next slide, in many cases, these variants may have only been reported in one patient or one family. So it's extremely heterogeneous. And looking in ClinVar, this is data just from this morning, actually, there are almost 1,600 GAA variants in ClinVar. And you'll see here about a third of them are actually VUS, and then 9% have conflicting classifications in ClinVar. So part of the goal of our expert panel was to try some of these as well, if we can. So as I mentioned, there are some so-called common pathogenic variants in GAA that are very well known and known to be disease-causing. And I'm showing these here. And of course, these tend to have different frequencies in different populations. So just good to keep these in mind as well. I do want to point out this one, which is a leaky splice site variant. So this variant does actually result in production of a small amount of normal GAA. And so these patients, anyone who carries this variant along with another pathogenic variant, will have late onset Pompe disease, because this is somewhat protective. There's enough activity of the enzyme to protect them from the infantile onset form. So another thing I wanted to mention in terms of why variant interpretation is important for this gene is that in the last version of the secondary findings statement from ACMG, GAA and Pompe disease was actually added to the secondary findings list. And this would be for a patient who had two variants that are pathogenic or likely pathogenic. And the reason that they state for adding this to the list is the availability of effective enzyme replacement therapy in infantile and later onset cases. So again, important to have variant interpretation for this reason as well. So just a couple of things before I move on to the specifications for the group. I did want to talk a little bit about some of the lumping and splitting issues for GAA. So not too complicated, but when you look in OMIM, there's only one phenotype, which is glycogen storage disease type 2. As I mentioned, this is, of course, a synonym for Pompe disease. In Orphanet, they mentioned the infantile onset and late onset form. And then in Mondo, you'll also see the infantile onset and late onset form under this umbrella term of glycogen storage disease type 2. So when we did the gene disease clinical validity, it was curated under this umbrella term. And the reason for that, as I mentioned already, is it really is a spectrum of disease. And of course, if you have, for example, one severe variant and one less severe variant, you're more likely to have late onset Pompe. But if you were homozygous for that severe variant, you're going to have infantile onset Pompe. So it really just is this spectrum, and the presentation is going to depend upon the variants present with some modifiers too. But in general, there's that correlation between the severity of variants, the enzyme activity, and then the presentation. And then just to mention that not surprisingly, the gene disease clinical validity was definitive. So I just wanted to mention that from the outset. So looking at the history of our VCEP, it's actually amazing to see how long we've been around for, but the group first got together back in spring 2017 and started developing or submitted our first application in 2018. We did our specifications that was submitted and approved by 2018. We went through our pilot. And so our first version of our specifications was approved in August 2019. And then in June of last year, we had submitted version 2. So I'm going to talk a little bit about our updates from version 1 to version 2. And as of a little before Christmas, we have 174 variant classifications in ClinVar and a few more in the eRepo. So the first thing I wanted to talk about a little was just the codes that we did not use. So we did not include either of the de novo codes for this disorder. As I mentioned, Pompe disease is a recessive condition. We very rarely will see de novo variants in patients, although a couple have been reported in the literature. But I think we just feel like it's not really a mechanism of disease. The mechanism of disease is loss of function. And it's just hard, even if you see a de novo variant, it's hard to kind of add additional evidence to this actually being the pathogenic variant. So we just decided not to include this. We rarely see them, and it didn't really seem to add any additional information to the pathogenicity. We also don't use PS4, as this tends to be a code that's more used for case counting for autosomal dominant conditions. Instead, our case counting code is more the PM3, which is the intrans code. I'll talk a little bit about that one. We also do not use the segregation codes, either for benign or pathogenic, and that was on the recommendation of the SVI. There is only one gene, as far as we know, involved with Pompe disease. And so any variant that you look at could certainly be in tight linkage with another variant. And so even if the variant you're looking at is not truly the pathogenic one, you might then have some sort of spurious results if you're looking at linkage. And then these other codes too just didn't really apply to our disorder. See, there's a question in the chat. Yeah, so from Deb, thanks Deb. If you have one inherited and one de novo, how is that handled? So we would not, I think, it's a hard one, whether you would know if they're in trans or not, but we wouldn't use the de novo codes, Deb. We would just look at that de novo variant.
So it would reach pathogenic.
Like in silico analysis, is there functional evidence? Okay. That type of thing. I think part of it is that, with autosomal dominant disorders, if it's a de novo variant and the patient has it and neither parent has, you have that kind of clinical information. For a recessive disorder, you would have to sort of look at something like enzyme activity, and sometimes there can be overlaps for this disorder. So it's just a little bit of a sticky kind of type of evidence to look at, we feel like, for recessive conditions. Okay. Is the recommendation not to use PP1BS4 applicable to all autosomal? I believe so. That would be more of a question for SVI. I don't know if Stephen is on, but my understanding is that if the disease has a single gene etiology, that you probably don't want to use the segregation codes, but I can confirm that with SVI. Unless it's Stephen on, I don't know. Okay. Excuse me. Okay, so moving on. All right, so beginning with some sort of straightforward information. So PVS1, of course, is a code that we will use, and we follow the recommendations from the SVI for PVS1. And we know, based on all the information that we have, that loss of function of this enzyme, acid alpha glucosidase, is the mechanism for Pompe disease. So there are, literally tens, hundreds of different loss of function variants in this gene, including some that are extremely well known at termination frame shift variants. So clearly loss of function mechanism, and of course also knowing from functional studies as well that the enzyme just doesn't work in patients with this disease. So as if you're familiar with this paper, you'll know there's a big flowchart within this paper that gives guidance on how to look at different variants. So I'm not going to go through every single PVS1 evidence that we use, but just to talk a little bit about this, for non-sensor frame shift, we just set our kind of cutoff at base 2749, which is about 50 base pairs into the penultimate, or from the 3' end of the penultimate exon. So anything beyond that, we would say, is probably going to be missed by nonsense mediated decay, and anything 5' we would expect to be picked up by nonsense mediated decay and make no protein. So anything beyond this nucleotide, you would be losing less than 10% of the primary amino acid sequence. And we don't really have a good idea of what that C-terminal end is doing. So we were sort of comfortable just using SVI's guidance and applying PVS1 moderate. And then I did want to mention initiation codon variants because, again, in the paper, the SVI is recommending using PVS1 at either moderate or supporting, but we had, a certain degree of evidence that made us kind of supported going up a little bit in strength. And I mean, you could think this is somewhat of a circular argument, but we do know that patients with initiator codon variants don't make any GAA protein, and there are multiple patients that that's been shown for. For example, Dr. Bali's group at Duke does a lot of work looking at cross-reactive immunological materials, blots on fibroblasts from these patients, and finding that there's just no GAA protein is made. Also, the next in-frame methionine is at position 122, but using predictors, the likelihood of the start site being used is very low. And even so, the gene product would be missing the signal sequence, so presumably wouldn't make it to the lysosome. And then when you look at some different initiator codon variants in terms of their functional activity, there's basically no enzyme activity as you would expect if you make no protein. There is a very faint band that's found on these Western blots, but based on Dr. Bali's work, this is thought to be simply a non-specific antibody binding. So for any of these initiator codon variants, we've been applying PVS1 at the strong level, and then of course looking at the other evidence as well. So that was our thinking there. So moving on to allele frequencies, and it was interesting actually, we had a conversation about allele frequencies for recessive disorders in our lab meeting the other day, and it really is challenging. I'll talk through some of the challenges here. We certainly did our best with the information we had, but definitely curious to hear others' thoughts on this too. So for BA1, we really wanted to set it conservatively high just to make sure that we didn't exclude anything that could be pathogenic. And I guess similarly for BS1, but a little bit lower, of course. And then we used much more strict criteria for PM2. So you have to be really pretty rare in order to meet this criterion. I did want to note as well that the recommendation from SVI is now to use PM2 at the supporting level as of September 2020. So initially when our specifications were approved, we were using this at the moderate level, but now we're using it at supporting. So like many other VCEPs, we're using the WIFIN calculator, the CardioDB calculator to set our allele frequency thresholds. So looking at the prevalence, the maximum allelic contribution, the maximum genetic contribution, and also the penetrance of the disorder. So I think this was our first challenge. What is the prevalence of Pompe disease? As I mentioned, most of the literature will state about 1 in 40,000, but it really does vary a lot across different studies. Certainly the infantile onset seems to be more rare than the adult onset, maybe about a third of cases infantile onset compared with late onset. A lot of variation across different populations, and also, of course, depending on the methods they used as well. And I see I have another message in the chat. Do you think some classifications will drop? That's a great question. And I know that Stephen Harrison has done a lot of work on looking at whether classifications drop from LP to P because of using PM2 at supporting. I don't know if Stephen is on the call. So we didn't do a lot of analysis on this for our specifications. We changed a number of them when we went from version 1 to version 2, and things kind of balanced out. But yeah, this is a great question, and I will ask Stephen about this.
Can I
he had done a lot of work on that, unless anyone else on the call knows on the fly.
I can just add that, that has been an issue for some of the groups in the hereditary cancer domain, and they have gone back to look at different codes that they can kind of boost, for instance, like other clinical data codes or phenotype codes that could be kind of increased to kind of counterbalance that. But yes, I think dropping PM2 to supporting does cause at least in several instances, that variants go to VUS, and they shouldn't be at VUS. So,
yeah, and that's a great point, Deb. So, like I said, we our codes, the changes we made kind of balanced out. We actually reduced the strength of functional evidence as well, but we upped the strength of our PP4 codes, and also that allowed us to use a lot of extra data. I'll talk about that a little bit, but yeah, I think it's all sort of very much in the balance of the different evidence that you use. And I apologize, I realized I had my sound switched off, so I apologize if anyone was asking questions before. I see another question in the chat. So from Kristen, that's been the case for our VCEP monogenic diabetes, no exact numbers, but we agree with the logic behind downgrading. Okay, thanks for that. Yeah, so like I was saying, just determining what the prevalence is of this disorder is a little tricky. And so this is from patients with a diagnosis of Pompe disease. But when the first data started to come out from newborn screening, this is actually a fairly old slide, you'll see that the numbers are much higher. And so there's various reasons for this. I mean, certainly it's quite possible that patients with later onset Pompe disease or even infantile onset just aren't getting diagnosed. So the diagnosed numbers are fewer than the actual numbers. Also possible, of course, that newborn screening, maybe there are some false positives, especially for those late onset cases. Maybe it's not as penetrant as we think. So we really just don't know at this point, but certainly the newborn screening numbers were higher than in the previous literature. So what we did then was in thinking about plugging some of these numbers into the Whiffen and Ware calculator, we used a higher number for BA1 and BS1 just to sort of give us that cushion, like I said, so we're not kicking out pathogenic variants. And then we used 1 in 40 for PM2. And then maximum allelic contribution, again, was kind of a tricky one because these variants are different frequencies in different populations. So again, it was somewhat of a sort of guessing game. But we used 1 for BA1, which is somewhat crazy, I guess, because we know that Pompe is caused by a lot more than just one variant, but that was sort of the most conservative estimate. It would give us the kind of highest for BA1. We used 0.5 for BS1 and 0.2 for PM2, which seemed sort of reasonable given the figures that we were looking at here. But again, somewhat of a estimate. Maximum genetic contribution was a little easier because there is only one gene for Pompe disease. Certainly, there could perhaps be some other factors that could affect the activity of the GAA enzyme. But as far as we know, one gene, so we used 1 for maximum genetic contribution for all of the codes. And then penetrance, as far as we know, it's fully penetrant. We do certainly know that there are hypomorphic variants where someone could be perhaps extremely mildly affected. But for now, we're using penetrance at 1. And I think it would be really interesting to see actually in the decades in the future what the newborn screening data shows. So these patients who were diagnosed at birth by newborn screening and what ends up happening to them in terms of clinical symptoms and so on, or whether some of them will just never develop the disease. But for right now, this is the best estimate that we have. And so plugging all the numbers in, we for BA1 said anything with an allele frequency above 1 would meet BA1. Between 0.5 and 1% BS1, and anything less than 0.1% would be PM2. And I should point out that some of the really well-known pathogenic variants that I showed in that earlier slide actually don't meet our PM2 cutoff, but we sort of created a PM2 exclusion list that said, like, we know these are pathogenic. We're going to put in still all the PM3 data and other data for those those variants, and they still met pathogenic. So that's kind of how we handled that. But I think that's another question that VCEP'S are dealing with as well. So as I mentioned, our case counting code is PM3, which I'm sure everyone who's looking at recessive disorders is using. So we follow the SVI recommendations and the point system, which I find is just super helpful, really nice just to have this points system. Of course, when you're looking at recessive disorders, often you are looking at compound heterozygotes. So you're kind of analyzing one variant, and then you've got this other variant, which is either in trans or you know it's in the patient, and you might not know the classification of that one. So that's always you know, a little bit of a challenge. What do you do there? So we would typically curate that second variant as well, so that we can decide how we want to apply these points. So one thing I wanted to talk about from the SVI recommendation for PM3, they clearly state that to avoid circularity in all instances, so whether the phasing is confirmed or unknown, the classification on the variant of the other allele should not use evidence from the variant being interrogated. So we've sort of taken this fairly seriously in our VCEP, and I'm going to show an example of kind of how this can work. So let's say that you're curating a case that's compound heterozygous for variant 1 and variant 2. Variant 2 is being classified. You already know that variant 1 is likely pathogenic without using data for variant 2, so that you can then use that as evidence for variant 2. Let's say that with all the information you have about variant 2, it reaches likely pathogenic. And you're like, well, maybe I can now use variant 2 to support variant 1. And that now makes variant 1 reach pathogenic. And now you can use that for variant 2, which also reaches pathogenic. So you can see there's this kind of circularity here, the circular logic, which is erroneously raising the pathogenicity of these variants. So this is something that should not happen. This is what the SVI is kind of just making sure that we are cautious about. So we're really careful in our group just to kind of keep track of which variant is supporting which, and make sure that the evidence only goes in one direction and not the other. And I do want to point out all the work that Jen McGlan did on this circularity issue at the beginning of our VCEP, and really sort of thinking about how this all works. So one of the ways that we've kind of dealt with some of this is to create a list of our top 20 non-loss of function variants. So these are sort of well-known pathogenic variants that we know are pathogenic. And if we see one of those in trans with a variant that we're curating, we can just automatically say, we know this is pathogenic. We know that the evidence to get it to pathogenic was usually using in trans data from loss of function or homozygous cases where possible. And so we kind of know where that evidence has come from, and just to kind of avoid that issue of circularity. And I do want to show this slide. I know some other curators at UNC have much more complex figures than this for PM3, but this is just really showing the way the evidence went for a bunch of the different well-known pathogenic missense changes. So you can see it can really get quite complicated. So we do have a project at the moment that's trying to track all this and just see how big an issue circularity is in PM3. And so I will report back on that when we have some more information. Okay, here's another question. Some of the older papers do not address the phase of the variants. How do you address this issue? Did you use SVI recommendations strictly? Yeah, so a lot of papers don't address the phase of the variants. I find, even if a table in a paper says variant 1, variant 2, you can't assume that they're actually in trans. I mean, I think authors often just make that assumption it's a recessive disorder. The assumption is they're in trans, but we would never make that assumption unless they say for sure the dad was tested and had this one, the mom was tested, had this one. So if we go back and look at the SVI recommendation, it doesn't actually require that the variants, excuse me, I'm just going back. Yeah, it doesn't require that they're confirmed in trans. If phase is unknown, you can still apply points there. So that's what we typically do. If it's a pathogenic variant, but the phase is unknown, we would give the 0.5 points. So, but yes, I find for Pompe, more often than not, they don't confirm the phase. Just keep in time here. Okay, so that's just a quick walk through some of the issues we had with our version 1 specifications, but actually around the time that version 1 was approved, there was this paper came out from SVI with Sarah Burnage as the first author, regarding functional studies. And I'll talk through our work on that, but we had initially used PS3 and BS3 at the strong level. And based on this paper, we realized we couldn't really do that anymore. We've also talked about PM2, so we downgraded PM2 to supporting. And then we also looked at PP4. I know Deb had brought that up as well, because we felt that our PP4 was really way too strict and we were throwing away a lot of data because of that. And then we looked at some other criteria as well, just to see, just to make some other updates. So for functional studies, as you may be aware, this is PS3 and BS3. So these are basically well-established in vitro or in vivo functional studies supportive of a damaging effect on the gene product. So we, again, based on SVI recommendations, are only looking at in vitro. We are not looking at enzyme activity in a patient cell. That is part of our PP4 criteria. I'll talk about that later. But these are in vitro studies where basically they have introduced the variant by site-directed mutagenesis and then expressed that in some cell type like a COS cell, AGK293, or GAA deficient fibroblasts, then measure the activity of the enzyme, and then often are doing Western blot as well to look at the synthesis and processing of the enzyme. So GAA is synthesized as this sort of proenzyme. It then gets cleaved, and then the active form is the 76 KD. So this is the wild type pattern. All of these are missense changes, and you can see that they typically will get stuck at this kind of pre-enzyme stage. And then often, of course, for the loss of function variants, you just won't see anything because those are picked up by NMD. So there are a couple of papers with lots and lots of functional data from a group that's really well recognized, well known in the Netherlands. And they published these studies where they had developed a really nice sort of system for looking at how well or how a variant impacts the enzyme activity. So they're looking not only at the activity of the enzyme, but also at the Western blot pattern as well. So how it's synthesized and processed. So they developed the sort of point system, and then they came up with this classification with anything from A to D as affecting the function of the protein, and then E and F are presumably non-pathogenic or known non-pathogenic. So it is an in vitro system, may not be perfect, but these are the results that they had published. And I will note that all of these were analyzed because there were rare variants of uncertain significance. So that becomes important later on when we look at the Burnich paper, because I'm sure many of you have looked at this paper and thought about this regarding functional studies. But if we think about this for GAA, so typically when you look at papers that are reporting these types of results, they will include a wild type control and also something like empty vector or antisense or something like that. And they often do multiple replicates as well. But one of the things these papers are typically missing is these known pathogenic or known benign controls. So like I mentioned, those previous two papers, they looked at these variants because they were variants of uncertain significance. So we really don't have these sort of known benign and pathogenic. This was an issue, and would really only allow us to use this data at PS3 supporting. So one thought that we had was, can we look at the variants in these papers and just see, is there additional information that's available now that would allow us to classify them without the functional data, and hence kind of add our own positive and negative controls. So Donna Kanevy, who was a postdoc at UNC, did a ton of work looking at these. We found 20 pathogenic controls, 2 were benign, but this would still allow us only to use the data at the supporting level. And basically you would need more benign controls in order to, up the level to strong. And indeed, when you look at the impact, so Deb, I know you were asking, somebody was asking about how this impacts classification. So for PS3, you can see how reducing this strength of evidence from strong to supporting really does affect the classifications here. So, but on the other hand, they're obviously concerned about these types of studies. So we wanted to make sure that we were using the guidance appropriately. So one other thing we thought about, having reduced our strength of PM2 and PS3 was, can we increase the strength of other codes? So we looked at PP4. which is the specific phenotype code. And in our version 1 specifications, we had required that papers would actually state the enzyme activity that was found in the patient. So we required less than 10% in leukocytes, lymphocytes, or muscle, and less than 30% in cultured fibroblasts. So this just kind of reflects the difference in activity that we see in these different tissues. And we know that patients with late onset Pompe disease can have as high as 30%, or sometimes even higher in fibroblasts, but we wanted to be somewhat conservative there. And we could upgrade to PP4 moderate if there was confirmation that pseudodeficiency variants are absent. I'll talk a little bit about these later. But again, in a lot of papers, you just don't have that confirmation, so that was tricky. And then we also required that PP4 is met in order to apply PM3. And so often in papers, you just don't see this type of data. They may say the patient had Pompe disease, they had all of these features, but they may say GAA was deficient, but they don't give the exact percentage of residual activity. So we just couldn't use that data for PP4 or PM3. So we were throwing away a lot of data. So we thought that, we really need to look at this again. So as I've already mentioned, there are lots and lots of different pieces of information that could be supportive of someone having a diagnosis of Pompe disease, including the clinical features, especially for the infantile onset form. It is does present in a pretty specific way with this huge cardiomegaly, the extreme hypotonia, and so on. And looking at the cardiac features, deficiency of GAA enzyme. We've talked about the Western blot. There are some biomarkers that can indicate Pompe, not specific, but certainly are seen in individuals with Pompe disease. Muscle histology, and then of course our molecular analysis. We wouldn't use that as support here because that's a circular thing, but lots of different things that we can consider when thinking whether someone has Pompe or not. So we went back and completely revised our PP4 and created a point system where, depending on how specific we feel that a particular feature is, we would add a certain number of points. We go to PP4 moderate at the highest, not as high as strong, but moderate is the highest. So if you have 2 or more points based on the system, you can apply moderate. PP4 for 1 to less than 2 points. And then less than 1 point, then you just don't apply PP4. But you could still apply PM3 if things were consistent with the disorder. So this has really been working out really well for us, I feel like, and it has somewhat balanced the reduction in strength for PM2 and PS3. So it's been working really well. As I mentioned, I did want to talk a little bit about these pseudodeficiency variants. So these are variants that in the lab appear to create an enzyme deficiency, but patients who have them and are homozygous seem to do perfectly fine. So these are 2 really common pseudodeficiency variants in the Asian population, which come out a lot in newborn screening and certainly complicate newborn screening. And there's different laboratory techniques that can be used to kind of tease those out a little bit. And then this one, which is common in European populations. So we always sort of keep an eye open for these as well. And we would never apply PP4 if the only piece of evidence was enzyme deficiency and the patient has pseudodeficiency variants, because you just can't use that as evidence. You don't know what's creating the enzyme deficiency. So there are some really, pretty well characterized pathogenic variants that are often seen in cysts with these pseudodeficiencies. And for these situations, we're really careful just to make sure that there's other functional evidence for these variants, multiple patients, and so on. I'm just careful to make sure that these truly are pathogenic and the enzyme deficiency in a test tube isn't just caused by these pseudodeficiencies. And I think we're going to be doing a bit more work on pseudodeficiency in the IEM working group as well, because obviously Pompe is not the only disorder that's affected by this. And then another code that we added actually was PM1, because clearly there's going to be some residues within the enzyme that are really important for its function. And so in 2017, actually, there were a couple of papers that came out that were looking at the 3D, the crystal structure of the enzyme. And both of these really, they complemented each other really well and showed the same residues that are really important. So this is what we took into account for thinking about PM1 and which residues we could actually apply PM1 to if they are altered. And so I will go through all of this in detail, but we have now a number of amino acids which, if altered, we would apply PM1 because we know that these are important either in substrate binding or in the catalytic activity of GAA. And then lastly, we also added BS2, which is for recessive disorders observed in a healthy adult individual if they are homozygous. So this was kind of an interesting one. We didn't include this one initially because patients can have late onset disease, and you do sometimes see homozygotes for some of these milder variants in NOMAD. So we felt like, well, even if you see a homozygote, you can't say for sure that it's not a pathogenic variant. So what we decided was that if you have documentation that an individual who's homozygous has normal enzyme activity, then you can apply BS2. To be honest, I'm not sure how often we're going to see that, but it's a code that we added and seemed to make sense. And then lastly, one other thing we added to our specifications was that Splice AI will be used to analyze all variants, including exonic variants, just to see if there might be any potential impact on splicing. There are a couple of fairly well-known variants that are within an exon but known to affect splicing for GAA. Certainly a number of them are like in the last nucleotide, which maybe isn't too surprising, but there's a couple that are actually within exons and by RT-PCR have been shown to affect splicing. So we just always run this just to be on the safe side as well. And then just to review our submissions to date. So on version 1, we had 137 ClinVar submissions, and using version 2, just 37. And you'll see that the vast majority of our submissions so far have been frameshift and nonsense, which are kind of, I guess, no-brainers for this disorder. But we're really trying to figure out our functional evidence codes, so PS3 and BS3. So we kind of held off doing more missense changes until we had sorted all of that out and submitted our version 2. So I see another question. Will you be comfortable? Yes. So a question, would you be comfortable using Splice AI to apply PP3? Yes, so we have actually for splice site variants that are not +1, +2, -2, -1. Splice AI is the tool that we are using. I'm trying to remember our cutoffs. I think it was anything above 0.5 for PP3 and anything below 0.2 for BP4, if I recall. But yes, so that's what we would be using. And also VCEP as well is another tool that's in our specifications. Okay. And then I did also just want to spend a couple of minutes talking about literature searching, because I know that this is a topic that often comes up, and it can be challenging to find literature, especially if there's no case reports or just a couple. So just making sure that you're not missing things. So with Pompe disease, we are incredibly lucky that there's this great database from the Erasmus Medical Center in the Netherlands, which basically has looked through all of the literature. It provides us list of variants, the PMIDs where that variant is located, and some basic information about the patients. Once in a while, I'll find a paper that's not in there, but in general, this is a really great place for us to start. We also have a hypothesis GAA database where we've had undergraduate volunteers who are annotating the literature. As of now, we have over 1,300 annotations, so this has been really great. This has been ongoing for a few years, and at some times it's more busy than others, but annotations are accumulating in there. It's not a full annotation of the literature, so but we would always check that as well, and it's a very useful resource. Personally, I really like Mastermind from Genomenon. The unfortunate thing is for the free version, you only have limited searches, but I do like that, it will sometimes typically find, at least most of the literature. Sometimes it can be non-specific, but I do like this resource. And then Google Scholar, Google Litvar, and ClinVar, of course, will also provide PMIDs if the submitters have included those. And then I have PubMed alerts set up for if there's any papers that are published on GAA or Pompe, it will appear in my email. So these are the resources that we're typically using. And then our curation and review process, I know that some VCEPs are super, have a very standardized process. Recently, we have, I guess, become more organized with this, which is really nice now with the Google site for ClinGen. So at the moment, we are sort of focusing now more on VUS and variants with conflicting classifications in ClinVar. So now that we have our version 2 specifications approved, we've started to look at these. These variants are added to a spreadsheet on our Google site, and then curators will sign up. We found that this is sort of the best way because many of our curators are volunteers, and so we just in terms of what their other responsibilities are, we just want to make sure that people have the time to do things and don't feel obligated to be signing up if they just don't have the time. So they'll then sign up, curate the variant, indicate when it's ready for review, and then for straightforward variants like loss of function, for example, those are typically reviewed offline and would be approved by 3 core members. For more complex variants, those will be reviewed on calls and again approved by 3 core members. And we meet twice a month still. It may be that now we're kind of up and running again with our version 2 specifications that we might be able to meet less frequently, but this has been working so far. And then our future work, continue our classification of variants in GAA, of course, and then we are beginning specification of rules for IDUA for MPS type 1, which is exciting, so looking forward to that as well. And of course, just want to mention all of our membership, the folks who are working on GAA. I already mentioned our co-chairs who are just fabulous, really great to work with, and our terrific coordinators, Meredith Weaver, who's been with us since the start, and Raquel, who joined more recently, but they just do a great job of keeping track of everything. We have all our great experts and curators, past and present, and then our previous members. So it really has been just a terrific group to work with. So that's it. I realize we have 5 minutes left and I flew through a lot of stuff. So please feel free if you have any questions to go ahead and ask. I don't think there's anything else in the chat that I saw. So any other questions?
Hey, Jenny, this is Kansana.
Hi.
Can you hear me properly?
Yes, I can. Yep.
Okay, great.
Hi.
So I know that PS1 is typically reserved for a different nucleotide change resulting in the same amino acid change.
Yes.
But I just came across a variant which occurs at the last nucleotide of exon 2. So it's a synonymous change. So it's not an amino acid change, but a different nucleotide at the same position has also been curated or classified to be pathogenic. Would you be comfortable applying PS1 for synonymous variants at the same nucleotide position, but with a different nucleotide change?
That is a great question. And I was hoping that Stephen, I don't see Stephen on. So we have not done that. So are you talking about GAA? Because I know there is one, that 546 position.
Yes, that's the one exactly I'm talking about, yeah.
So yeah, I think there are 3 different changes at that position, if I recall. So we can apply PS1 for that, I think, because it is a synonymous change. And so far, we've only been using PS1 and PM5 for missense changes. And I think that's sort of the more conservative approach. I would be curious to hear from SVI, what the thought is for that. I know I think some VCEP'S might be using it for splice site changes. I don't know if anyone here uses PS1 or PM5 for different intronic changes at the same nucleotide. Kristen says we use PS1 supporting. So Kristen, that's for if it's for splice site variants at the same nucleotide. So yeah, I mean it seems like so some groups are using that, and I can see. I think sometimes groups will require additional things. Like so, for example, if it's creating a difference in splicing, you may want to see like a higher splice AI score for the variant you're looking at compared to what is pathogenic, if that makes sense.
Yeah, that's what even Kristen says, right?
Yes.
Great. Yeah, thank you.
These are really great questions. Yeah. Any other questions?